Hello friends, this video is on membrane proteins. It comes in very first chapter of physiology, the composition of the cell membrane and the various constituents. So let's start from the basics. The cell membrane has a typical composition like this. It has got carbohydrates, lipids and proteins with occupying certain surface area. For example, carbohydrates 3% surface area occupied, lipids in the membrane they occupy 42% surface area, and proteins occupy 55% of the surface area of a typical membrane. And molecule wise they exist in a ratio of 1:1. The lipids and proteins, they would exist in the ratio of 1:1. So this is a very typical composition of a cell membrane. However, the point that should be noted here is about the membrane proteins. We have already said that proteins occupy about 55% area of the membrane, and therefore we are by that we imply that there is certain fixed amount of proteins in a cell membrane, but that's not the case. The protein composition or the fraction of proteins or the percent of proteins would vary from cell to cell, or let's say membrane to membrane. For example, in certain membranes, for example, inner mitochondrial membrane, there is 76% fraction is of proteins. So very high protein content. Or let's say presynaptic membrane. It has got channels, and channels are proteins, and therefore, and it is devoid of any myelin sheath. Myelin is high in lipid. So therefore, protein content is more, relatively greater protein content in the presynaptic membrane. So the point is that a protein composition or protein percentage varies from cell to cell or membrane to membrane, point number one, and it varies for a particular membrane also. It will vary from time to time. Yes, isn't that surprising? Although we have discussed that a typical cell membrane will have a certain amount of protein, let's say 55% area occupied, etc. But it's not a very fixed ratio or a fixed percent. For any typical membrane also, its protein composition or protein percentage will vary from time to time. Yes, what I'm trying to suggest is at some time the protein composition may be higher than usual. At other times it may be lower in a particular membrane. So for a particular membrane, how the protein composition can change from time to time, let's see that, and that's the topic for discussion in this today's video. So the protein composition or protein percentage for any particular cell membrane, it depends on a particular factor called as turnover rate of the proteins in that particular cell. Membrane proteins will have a certain turnover or turnover rate, and that will determine at any given moment, at any given point of time, how much is the protein percent in that particular cell membrane. And of course, at the end we will also discuss a Physio plus plus in the form of application of this topic, and that is in the form of aldosterone and how it increases the sodium reabsorption. So let's understand what this turnover rate means. Look, proteins are synthesized inside the cell. So membrane proteins also, just like any other protein, would be synthesized within the cell. And then there would be certain rate of synthesis, obviously. Then the membrane protein will be brought to the membrane. It will be anchored to the membrane or fixed to the membrane, and then it will be removed from the membrane. It will be endocytosed. It will be taken back into the cell, and then it will be degraded in the cell organelle. Generally, the membrane proteins are degraded in a cell organelle called as 26S proteasomes, or some proteins are also degraded in lysosomes. However, this is the turnover rate for a particular protein, membrane protein or otherwise any protein also in general. Synthesis, then going to its targeted location, and then after its lifespan, it will be removed from the membrane and degraded. That's the life cycle and that's the turnover. So for example, if the rate of synthesis for a particular protein is higher than the rate of removal, then what will happen? The membrane protein composition will increase, obviously. On the other hand, if the rate of removal of that protein, the rate of degradation of that protein is higher at any given moment, then the membrane protein composition or percentage will drop because the proteins are getting degraded faster. So that's what is meant by the protein turnover rate. And let's now understand what determines this protein turnover rate. Here we need to understand a particular system in the cell which is called as the ubiquitin system. Now, as we have mentioned just now, that the membrane proteins, once they have served their function, once they have served their lifespan, they will be removed and then they will be endocytosed, taken back into the cell and then degraded in the cell organelle. Before they could be removed and degraded, they are first tagged that this protein is to be removed. So this protein will be tagged, and membrane proteins are tagged for removal or replacement by this particular system called as ubiquitin system. So that's the first important point to be recognized, that let's say here is a protein, here is a transport protein in a cell membrane. Now this, if it has served its function, then it will be tagged, of course, from inside the cell, from inner aspect of the cell. It will be tagged because the ubiquitins are, of course, inside the cell. It will be tagged by the ubiquitin system for removal. Ubiquitin is not degrading the membrane protein. It is only tagging the membrane protein for the removal. Okay, so this protein has to be removed now. Ubiquitin will tag this protein for removal or replacement, whatever the case. And then, before we proceed, of course, we have already said that once it is tagged, then it will be removed from the membrane and it will be degraded. So let's understand a few more things about ubiquitin. Ubiquitin has a system of enzymes. Basically, ubiquitin is a 76 amino acid protein and attaches to the membrane protein by a particular enzyme or a particular family of enzymes called as ubiquitin protein ligases. So there is an enzyme, or there's a group of enzymes called as ubiquitin protein ligases. By these enzymes, the ubiquitins will be attached to the membrane protein. Ubiquitin protein ligases will attach the ubiquitin to the membrane protein. And here's an example. NED4 like family. It's a family of ubiquitin protein ligases. The other point is cells also contain DUBs, that is deubiquination enzymes, deubiquinating enzymes. So if the protein is tagged by ubiquitins, but it has not to be removed yet, it has not to be degraded yet, it has not served its lifespan, then there is deubiquination, means ubiquitin will be detached. It will be made, this protein will be made free of ubiquitin so that it can continue to function for some more time. So the point I'm making here is there are two systems under the ubiquitin. One is ubiquitin protein ligases, which will bind the ubiquitin or attach the ubiquitin to this membrane protein so that it will be tagged and removed from the membrane. And there is a system of DUBs, deubiquitinating system. So ubiquitin will be detached from this membrane protein, so it will not be removed, at least for some time. It will not be removed from the membrane, it will stay on, it will not be degraded. That's a fine balance between ubiquitin protein ligases, which attach the ubiquitin, and DUBs, which detach the ubiquitin. All right. Now, amount of protein in a cell membrane at any given point of time depends on the rate of addition of ubiquitin groups by the ligases versus the rate of removal by the DUBs. That's the key point that you need to understand here. That there is a rate at which the ubiquitins are getting bound to the membrane protein versus the rate at which DUBs are acting and removing the ubiquitin from the membrane proteins. So, amount of protein in a cell membrane depends on the rate of addition of ubiquitin groups versus the rate of removal of ubiquitins by the DUBs, and that will finally determine how much membrane protein is there in a cell membrane at any given point of time. So that being said, I hope the point has been highlighted very well, that a membrane protein composition and the protein protein percent in a cell membrane at any given point of time will depend on this kind of turnover. Right. Now, that being said, let's now talk about the Physio Plus Plus, the application of this in the clinical physiology. And the application is in the form of mechanism of action of aldosterone. You must have read this, that the aldosterone increases the sodium reabsorption from various places, but predominantly from the kidney. It performs this function elsewhere also in the body, but predominantly in the kidney, aldosterone increases the sodium reabsorption, and because of that, water reabsorption also increases. So how is it that the sodium reabsorption is increased? Here is a typical diagram of sodium reabsorption from a segment of the nephron. Sodium in the tubular fluids in the lumen, and here is a lining cell. You can see the cell lining the tubule. Now the cell has two sides, as you must be aware. The apical side, apical membrane, which is facing the lumen, and the basolateral side, which is facing the blood vessel. And these two sides have certain transport proteins. So we are talking about the membrane proteins in the form of transporters, transport proteins in the membrane. So how does it all happen? On the basolateral side, in the basolateral membrane, there is sodium potassium pump. And you know what is the function of sodium potassium pump? To remove the sodium from inside the cell and send it out. So sodium will be sent out of the cell, and finally it will be sent into the blood. And in the apical membrane, there is another channel, another transport protein called as ENaC, epithelial sodium channel, epithelial sodium channel. So sodium reabsorption occurs in a two-step process. Sodium from the tubular fluid in the lumen, it will first enter the cell through the apical membrane via this membrane protein, transport protein, the ENaC, which is a channel, epithelial sodium channel. And then from once inside the cell, then it will be removed from the cell and sent into the blood by action of sodium potassium pump. So we have two membrane proteins. Here essentially we are talking about two membrane proteins. One on the apical membrane, that is ENaC, the other one sodium potassium pump on the basolateral membrane. And our topic is related to the membrane proteins. Now, aldosterone increases the sodium reabsorption. We have already said that aldosterone increases the sodium reabsorption. How? By increasing the stay of ENaC in the apical membrane. ENaC, it's a membrane protein, and whenever it has served its function, this ENaC will be tagged by a ubiquitin system called as NED4, or to be more precise, NED4 slash or hash 2, NED4 2. Now this is a ubiquitin which will tag the membrane protein, this membrane protein, the ENaC, so that the ENaC is removed from the membrane and then degraded inside the cell. Aldosterone acts on this NED4.2 or 4-2, and it inhibits this ubiquitin. Aldosterone inhibits the ubiquitin NED4-2, which means what? If the ubiquitin was inhibited, means now the ENaC will not be tagged, and if it is not tagged, it will not be removed from the membrane, and it is not removed from the membrane means it is not degraded. So ENaC will stay for longer duration in the membrane, which means the number of ENaC proteins will be higher at this moment as the aldosterone begins to act. Okay, so ENaCs staying for longer duration and therefore their number temporarily higher in the membrane. And on the basolateral side, there is sodium potassium pump. So aldosterone is known to increase the number of sodium potassium pump also. Increase the number of, aldosterone is a mineralocorticoid. It has got a receptor called as mineralocorticoid receptor, or MR. That's a topic for another video. But it has got a nuclear receptor, and that means it can increase the number of protein, or it can increase the protein synthesis. So it increases the number of sodium potassium pump also in the basolateral side, on the basolateral side. And that is how aldosterone increases the sodium reabsorption. It is increasing these two transport proteins in the membrane. And how? By acting on the ubiquitins, by not allowing the ubiquitins to remove this ENaC from the apical membrane. So that's the application of the knowledge that we have about ubiquitin enzymes, and the application in the form of action of aldosterone and how it increases the sodium reabsorption. I'll be continuously or rather regularly be uploading these videos explaining short concepts. So what are you waiting for? Subscribe to the channel.